Dr. Saporta, you're back again with us. As was Dr. Herman, you were both here yesterday, and we're really grateful for the time that you're giving to us, both the scientific community in our meeting yesterday and then the CMT patient community today. So thank you. Dr. Saporta is at the CMT Center at the University of Miami, where he also teaches neurology and human genetics. His expertise lies in the diagnosis and treatment of CMT. I'm seeing a lot of questions come through about diagnosing. I want to remind everyone that in our Meet the Expert session that opens up at 3:15, you'll have the opportunity to meet with Dr. Saporta, and you'll also have the opportunity to meet with Dr. Arshana Feely, who's a genetic counselor. So you can ask some of those very pointed questions to them then. But still continue to add your questions into the Q&A box and we'll get them answered for you. Back to Dr. Saporta. So he is the scientific advisor of the only phase 3 clinical trial in CMT, which is the FARNEX trial that a lot of you have been asking about today. He will also talk about clinical trials for CMT, past, present, and future. So welcome, Dr. Saporta.
So good afternoon, everybody. It's really a pleasure to be here again with the CMT Research Foundation Global Research Convention. As Susan introduced me, my name is Mario Saporta, and I work at the University of Miami. I just have a few disclosures to make because I'm going to be talking about the FARNEX Premier trial. I want to disclose that I'm the principal investigator for North America for the trial, and on that role, I am a consultant for FARNEX, and I also receive research funding from multiple patient foundations and also from biotech companies. So in the next 10 minutes, I'm going to cover those three major topics. One, it's really the focus of the talk, which is the ongoing trial for PXT3003, or the Premier trial for CMT1A, what we call a near future trial for SORD neuropathy. And then the future, which I think you all heard for throughout the day, the new therapies that are really finishing preclinical development and are really getting to a point where they're going to start first in human studies soon. So about the FARNEX Premier trial. So as you all know, this is a trial specific for CMT1A. The compound, it's called PXT3003, but it's actually a combination of three medications, all of them FDA approved and in clinical use for decades. Which is a muscle relaxant, sorbitol, which can be used as a sweetener or to treat constipation or gastrointestinal issues, and naltrexone, which is a weak agonist, but actually acts as a competitive antagonist, meaning it's used to treat patients with opioid addiction. So they're not being used for their primary indication, but through the network analysis that FARNEX has proprietary ownership, they determined that the combination of those three drugs actually were helpful in bringing down the levels of PMP22, which, as you all know, it's increased in CMT1A. So this drug has gone through a preclinical study in different models of CMT1A. It moved to phase 1 and phase 2 trials, which is really geared towards making sure that the combination of drugs is safe. We already knew that each individual drug was safe enough to be FDA approved, but because they are being now given in combination, you have to go to this process of confirming that the combination is also safe. And it actually went through a phase 3 trial a couple of years ago, but that trial had issues with the manufacturing of the high dose compound. They had problems with the compound forming crystals, and the European Union and then the US decided to stop the arm taking the high dose formulation. And even with the trial being a positive trial, meaning it showed benefit using the metrics that we're going to quickly talk about, the FDA require Pharmax to undertake a second phase 3 trial, which is the current trial, Premier. This trial is a 15-month study, meaning the study only ends when the last patient to be enrolled completes 15 months in the study. And it consists of 6 in-person visits. So patients have to be physically in the clinic to be examined by the investigators. And in between visits, monthly telephone calls to check how the patients are doing. The evaluations are actually fairly straightforward in terms of there are no invasive or complicated procedures. Patients are examined with a general physical exam and a neurological exam, and that will allow us to fulfill most of the CMT scoring systems that are used for this trial. In some of the visits, patients will also undertake a very limited nerve conduction study without the needle part, just a nerve conduction study. ECG and blood work for safety profiling, again, to make sure that there's no surprises throughout the trial. A walking test where we ask patients to walk as fast as they can for 10 meters, and we time that. And that's one of the secondary endpoints or the secondary parameters that we're going to be using to determine if the drug is helpful or not. And then the measurement of strength of the ability to move the foot up, which we call ankle dorsiflexion, and also of your grip using a specific equipment called dynamometer, which gives us a number for the strength. Patients will be divided 1-to-1, meaning half the patients will be receiving the high dose of PXT3003 and half the patients will be receiving placebo. The two compounds taste the same, they have the same consistency, so we'll be blinded, both the physician and the patient, to whether they are on drug or placebo. As I mentioned, the major endpoints or the major ways we're going to measure whether the drug is successful or not is the walking test that I mentioned and a scoring system called the Overall Neuropathy Limitation Scoring System, which is a general neuropathy scoring system. So it's really not originally designed for CMT, though it was validated in a mixed population of patients with different neuropathies, including CMT, and it really focused on patients' limitations caused by the neuropathy, both in the lower extremity and in the upper extremity. The target of number of patients is 350 patients in total, and we have 52 study locations that will be recruiting those patients, 25 in the United States, 24 in Europe, and 3 in Israel. We have sites actively recruiting, including us here in Miami. And to get more detailed information, you can get to clinicaltrials.gov, and that's the number that you can use to identify the study. But you can also just go to clinicaltrials.gov and look for Pharmax, PXT3003, CMT1A, and you should be able to find the trial. And there you'll find the most updated information of the trial. So just to give you an idea of where the centers are for the United States and Canada, here's the distribution. The green dots are marking centers that are already recruiting patients, and our 3 centers here in Florida are already evaluating and starting patients on medication. And then the red dot mark the centers that are still in the process of getting started, so they are not actively recruiting. As you can see, we have a fair amount of centers in the West Coast, the East Coast, and a few in the Midwest, and 1 in Texas, and then 4 centers in Canada. In Europe, we have centers in Spain, Italy, France, Belgium, the Netherlands, Germany, and Denmark, and most of the French centers and the German centers and the Spanish centers are all active. The Italian centers and Netherlands and Belgium is still kind of catching up. and Israel is also not active yet. But for patients that are calling from outside the US, those are the centers that are either actively recruiting or are going to be seeing patients for the trial. Just to give you an idea, not every patient with CMT1A would be eligible to be part of the study. For every clinical trial, we try to have a very balanced and what we call homogeneous or very similar population of patients, so that we can really avoid confounding factors or anything that would cause any imbalance that could generate a false result that's not caused by the drug, but by an unforeseen or an unexpected difference between groups. So for this trial, the inclusion criteria are patients have to be either male or non-pregnant females. The age is between 16 and 65. And they have to have genetically proven CMT1A. So in some trials, they accept to have a first-degree relative with a genetically proven diagnosis and patients with symptoms. In this one, every single patient has to have a genetically proven diagnosis of CMT1A. And in the patients that we know have CMT1A because they are the son or the brother or sister of a CMT1A patient and have the same presentation, the study itself offers the testing just to have the personal confirmation. The patient has to be able to provide written informed consent and comply with all the study procedures. They have to have mild to moderate CMT as defined by the CMT neuropathy score of more than 2, but up to 18 points. They have to have some degree of muscle weakness in foot dorsiflexion, though there's no formal limitation of the weakness, but if you're very, very weak, your CMT neuropathy score would probably exclude you from the study. You have to have a specific conduction velocity on nerve conduction studies and be on stable prescribed drugs with no expectations to change the dose. And if you're female and also male, you have to have or take specific care not to either become pregnant or have a child during the study because of the risk for the gestation of the medications. The exclusion criteria is pretty long, so I'm not going to go through all of them, but we do this check for every patient that we see. So of course, if the patient doesn't have CMT1A, he cannot be in the trial. If he has such a mild neuropathy that their overall neuropathy limitation score is 0, they are also not going to be able to be included. And if there's any confounding factors, other neuropathy, other diseases that could affect the way we evaluate patients, that also is an exclusion criteria. So that's for the PX33003, and I'll be happy to answer questions either in this session or in the talk to the specialist session at the end of today's meeting. I want to move to the next trial that we think will follow PX33003, which will be focused on this new CMT type called SORD neuropathy or sorbitol dehydrogenase neuropathy. This has been recently discovered, and it seems to be the most common cause of autosomal recessive CMT. And because it affects an enzyme in a very well-known pathway where glucose is processed in the body, we believe that treatments will quickly become available. There's a company called Applied Therapeutics, and they have compounds that could help treat this specific type of neuropathy. And we believe that this neuropathy, which is caused by the loss of this enzyme here, it's actually caused by accumulation of sorbitol in the peripheral nervous system. I know that sorbitol is used first on the PX33003 trial, but this has nothing to do with this other type of CMT. This one, sorbitol, it's bad. It accumulates and it causes damage to the nerves. So the ultimate goal in this neuropathy is to bring this chemical down. And one way to do it is to inhibit the enzyme that converts glucose into sorbitol. And that's what all those reductase inhibitors do. And this company has a compound called AT007 that they're testing in a small group of patients with SORD neuropathy to see if they can reduce the sorbitol levels of the patients measured in the blood. There's an information in their website. This is the email. So for patients that have been diagnosed with sorbitol dehydrogenase neuropathy or SORD neuropathy, you can contact the company because they are interested in recruiting patients for the phase 1 and the next phase studies that are upcoming in the near future. And the last slide for my talk is really to give an idea, and it's basically a summary of everything that has been discussed yesterday and today. It's really the amount of strategies that are now being advanced in preclinical studies. And when I say advanced, I mean that they're basically finishing that first step, which is showing that the approach works. So there are multiple ways to bring PMP22 expression down using different genetic tools, at least 3 different that we heard of yesterday. And all of them have been successful in treating animal models of PMP22. I believe that we are going to start seeing those strategies moving to clinical trials. Some of them actually are the same tools used for other neuromuscular disorders that have been now FDA approved for use and are being prescribed for instance, spinal muscular atrophy and TTR neuropathy, which is a different type of neuropathy than CMT, but it's also genetic. And there's a lot of work with CRISPR-Cas9 in a way to correct mutations in animal models and cellular models from patients with different types of CMT. So it's not only PMP22 where we're seeing a lot of work, we're seeing the same tools being applied for CMT1X, CMT2A, the recessive forms of CMT with gene replacement therapy. So there are studies for CMT4C, CMT4A, CMT4J, and we are expecting those to become clinical trials soon. And we know that in science soon means at least a couple of years, but I think we're very close to seeing a revolution in the treatment of CMT, and for sure in the number of clinical trials that are upcoming. And that's not to mention the other strategies that might have a even more kind of widespread effect in treating CMT. HDAC6 inhibitors are chemicals that have been shown to help several type 2 CMTs and also CMT1A. There are treatments for CMT1B that are also in the making, and also other gene replacement therapies. They are not gene specific, but they might help with different types of CMT. So again, a lot of things are happening actively in the preclinical arena that are in the verge of translating into clinical trials in humans. This is my email. If anybody has any specific questions, that's the best way to reach me. And thank you for your attention. And I look forward to talking to you later.